This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 971, recorded on December 30th, 2022. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dixon Despommier.
Hello, everybody. Looking out my window, we always do the weather, right? It's a gorgeous day. Really, for this time of the year, it's amazing. It's in the low 40s. There's no wind. The birds are all over my bird feeder on my porch, and so is everything else that birds produce after they eat. And my wife is very upset with that, but she loves watching the birds also, so we're having a good time. It's a wonderful time of the year.
Yeah, it was just earlier this week. It was bitterly cold, remember?
Exactly.
Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. 63 degrees and cloudy. Tomorrow's going to be 76 and sunny. I went for a walk around Lady Bird Lake this morning. It was very nice.
Lady Bird Lake, cool. Also joining us from Madison, New Jersey, Brienne Barker.
Hi, great to be here. Continuing the beautiful weather. It is 58 degrees here and sunny. Yes.
58?
Yes.
Oh, my word.
And from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it's 51 degrees Fahrenheit, which is 11 Celsius. And I am lamenting the fact that this is the last day I'll be able to do this with my favorite weather app, Dark Sky, because it's being subsumed by Apple. And it's not going to have the great user interface, I can already tell.
Too bad.
But it's raining, and it will continue to rain through the night.
That's too bad. Well, this is our last TWiV for 2022. It will be released on the 1st of 2023. And I don't know how long we've been doing this, but we usually do this episode as a review of the year in virology, right?
Yeah.
I don't think we did this from the beginning.
I think we probably did. We did it in year one.
How about that?
I'm guessing. I'll look it up while you're...
It's hard for me to remember.
What year is this now that we're in? I started in 2008.
Started 2009? 2008?
2008. September, so it's roughly, what? This is 2022, so that's...
That's 24 years.
No, it's not 24 years. It's 16
14.
years, right? No.
Come on, kids. We can do this.
I'm not even trying.
2022 minus... This is very embarrassing. 2008. 14 years, is that right?
14 years.
Okay, the first episode was West Nile virus.
Yeah. Yes.
September 24th, 2008.
8.
And let's see here. So we'd be looking for... December 13th is 11, 12, 13. Look at this.
I'm looking.
TWiV 13.
Right.
December 28th, 2008. Host Vincent Racaniello and Alan Dove. Top 10 virology stories of 2008.
Wow. Episode 13.
So I think that qualifies.
Yeah, it does.
Yeah.
We'd only been on for a couple of weeks, but...
Exactly.
13 episodes. Wow. Yeah, so I had to get Alan because Dixon disappeared.
He did.
He said, I'm going to India. You went to India for like 2 months.
I went to India? How could I have done that?
In fact, you left in the middle of an episode. You went out the door. I said, don't let the door hit you on the way out.
Oh.
Yeah, that's kind of a backhanded thing.
Yeah, we know what that means. We know what that means.
Anyway, so we do these annual reviews. They get more and more complex. And we kind of go over a bunch of things of the previous year. First of all, we went from 848 to 970, because this is always a thing, who knows? This one is recorded in 2022, but it drops in 2023. And so we'll count this as a 2023 episode, although it shouldn't be. 122 episodes, including 52 clinical updates with Daniel. Now, at the beginning of the year, we were doing 3 a week. Now we're down to 2. I think we went down to 2 in the spring. But in the previous year, 2021, we did 3 every week. So we did a lot more episodes then. And we had also 2 specials. TWiV special. Paul Offit and Daniel Griffin, an MPOX special. And so you may ask, what is a TWiV special? That's a good question. There's no manual of instructions. I tend to say a TWiV special is shorter. It's less than an hour, right? But those of you who are observant will note that the Carl Zimmer is less than an hour, and it's got a number. So it's whatever I feel like it. I don't know.
And the ones at meetings like at ASV, they're pretty close to an hour, if not less than an hour.
So they're not specials either.
So I think the specials are ones that you just feel like, oh, I don't want to give this one a number. I need to adjust the numbers so that we're going to come out on a good date for TWiV 900 or
I think
TWiV
you're right.
1000 or whatever.
Yeah.
The TWiV manual was written by Calvin, as in Calvin and Hobbes.
Right.
Yeah, if you're familiar with Calvin Ball, you'll know what
manual.
that's like.
Yeah, so the special is, like Kathy said, I don't really want to give it a number. So the Griffin, I said, Dan, let's do a special MPOX, and then it became a special. And then I think the previous year, there was a presidential special, right, when Trump got COVID. So, and then Offit was 30 minutes. So I thought, and we arranged that just the week before. Anyway, it's just something that doesn't need a number. But often, if you notice, the interviews, if I do on the road, I'll call them a special TWiV because I don't know when it's going to be released. But then when it's released, I'll give it a number, like Taubenberger and Zimmer and so forth. Okay, so that's that year's productivity. We have a new host this year. Angela Mingarelli joins. She'll be doing 1 a month with us because she's a PhD student, and that's not a bad workload. So we're looking forward to having her. I was gonna say to her, hey, why don't you come on for the year's review? And then I see on Instagram she's in Costa Rica, so can't make it.
So I went to India. She went to Costa Rica.
Yeah, she's in some beautiful forest.
They have great.
Yeah, I've never been.
Maybe she'll see the glass frogs that I talked about last week.
It made the cover of Science magazine, for God's sakes.
Very good.
Did you see that?
I did.
I didn't, but I believe you.
Well,
We had 47 unique guests. Unique means some people came 3 times, like Paul Offit. But we had Alessandro. I'm going to read them all because we thank them for coming. Alessandro Sette, John Udell, John Mascola, Michael Waraby, Richard Plemper, Elke Mühlberger, Adam Hume, Gabor Kemeneshi. He told me you have to have the shh at the end. Paul Offit 3 times. Vincent Munster, Raina Plowright, Pamela Bjorkman, Andy Meal. Is it Meal or Mealy? Mealy. Michelle Banks, David Baltimore, John Coffin, Harold Varmus, Kartik Chandran, Denise Hasselwanter, Gorka Lasso Cabrera, John Dennehy, Mark Johnson, Davida Smith, Monica Trujillo, Nels Eldy, Jane Hartman, Andy Slavitt, Michael Merchlinsky, Cedric Fischott came twice, Emma Thompson. I'm reading these. I have memories of each of them. It's really cool. Tom Monath, Eddie Holmes, Lori Garrett. So Lori actually had her own with us. And then I did a separate thing with her, and I released it in the TWiV feed, but I didn't call it a TWiV. But I did that so that people would watch it. Catherine Hanley, David Quammen, Richard Knight, who has, I think, the best intro. Before we started recording, I said to the audience, Richard just gave a talk, and you may hear some of the same questions, but that's all right because this is being recorded. And then he said in kind of a sotto voce, you might hear different answers though. And so I left that in as an intro because it's so funny. The audience really laughed. Jake Scott, Michael Letko, Anthony Frank, Dan Wilson, Chiara Mingarelli, and then Angela Mingarelli came on first as a guest. Gary Whitaker, Esper Callis, Jeffrey Taubenberger, Carl Zimmer, and then Matt Rain, who is our savior from vaccinated.us, who's given us many months of Spike Shirt profits. And he came on for 20 minutes to just talk about what he was doing there. So, it wasn't a full guest, but he was a guest. So, and boy, we appreciate your help very much. So those are our guests. Thank you all. 47.
When I saw you read Jane Hartman, I remembered that you had Michelle Banks on the same episode. So that makes 48.
48 unique. Yeah, I don't know why I left Michelle off. Yeah, she was there. So that was an episode, interesting episode. It was supposed to be Jeff Taubenberger. He was at ASM Microbe. And his mother got sick, so he couldn't make it, understandably. So right next to the stage, there was the booth with Michelle Banks and Jane Hartman selling their art. So I walk over. I said, in an hour we do a TWiV. Can you two come? And they said, sure. And they got people to watch their booths. I think it was great that both of them did it, right? And I didn't have any questions. I scribbled a few questions down. And Michael Schmidt said, I'll come help you. It was a nice collaborative effort and a good episode. Oh, 48 unique guests. This is almost one a week, right? That's a lot.
Mm-hmm. Yeah.
Mm-hmm.
Even
That's
though we
a
have
lot.
some of them are multiple per week. So anyway, we'll have more guests next year, including starting in January. We have a couple lined up already because they're a lot of fun. And I think listeners like them a lot. I had a thing here that said how many COVID and non-COVID episodes, but I decided not to do that because we had a lot of non-COVID stuff. We tend to mix them up, though, in this year.
Yeah, there's lots of episodes that are part COVID, part
Hybrid.
not.
Yeah.
Yeah, it's hybrid episodes. So we could get in our virology fix and then people could hear about COVID because that's what they're here for. And so I would say the big stories in 2022 was COVID, of course, Mpox and poliovirus and a little bit of Ebola, right? Those are the big ones, wouldn't you say?
Yeah.
Maybe a little RSV near the end.
That's true because of the
RSV,
big
yeah.
numbers, yeah. RSV.
Vincent, how many downloads did we have total?
I don't know.
Millions.
A lot. Well, so the thing is we have YouTube and then we have audio only, right?
Yeah.
So the YouTube, every episode goes on YouTube.
Right.
We have 116,000 subscribers on YouTube. We just went from 115 to 116.
116,000, that's right. That's
very good.
remarkable.
So not every one of those watches every episode, but I would say the TWiVs get anywhere from 6, 7, 8,000. You know, the offits are over 60,000 views.
Yeah, yeah.
Right? So it depends. And Zimmer is doing very well also. I mean, I usually release them at midnight. This is interesting. And then I get up 6 or 7 o'clock. I look to see how they're
Right.
doing.
Oh, yeah.
And if it's less than 1,000, it's typical. It's going to get, but already at 6:30, Zimmer was 2,500 views. So I said, that's going to do well. Right? It's an
And David
interesting-
Corman must have done well, too, then.
You can go and look. You can see all the views there.
Yeah, I will.
And then there's the audio, which as soon as it releases on Sunday, it gets immediately 30,000 downloads, and then it can go up 50, 60, 70,000, depends on the episode. So,
Wow.
Those auto downloads are because of subscriptions. I take
many
it.
of them are auto downloads, so you really don't know who's listening to them. And this is we don't do ads, fortunately. So, but because if you buy ads, you have to guarantee so many downloads. But the ad people say, I don't know, who knows if you're listening, right? So Apple is trying to deal with that in their own way, but we don't. I'm happy not to do ads because I don't want to get into that business. Anyway, I noticed this week that Christian Drosten says the pandemic is over.
That's a
Yeah.
relief.
We're now in the endemic phase. So there you go.
Except in China.
Except in China, for sure.
Yeah, well, 1/8 of the world's population. That's not so bad.
I think, from a strictly academic point of view, the situation in China is really an amazing twist
Yeah.
on the whole
No
pandemic.
question.
I mean, I don't want to trivialize the suffering
No.
or the problem. Okay, but from an academic point of view, it's fascinating.
So
Well,
what's,
politics also. A lot
in
of
your
politics.
opinion, what is the reason why they're in this quandary?
inadequate vaccination policy during the shutdown.
Right.
Okay, and that includes a vaccine or vaccines that were not up to snuff, number
Correct.
one.
Correct.
And number two, and this is what really blows my mind, not enough people vaccinated. I would have thought in China,
Yeah.
Just line them up.
I mean, I guess I don't understand the interface between the politics and the culture in China. But I would have thought that in a place that can enforce a lockdown to the extent that they could, you would be able to get everybody vaccinated. Okay, so that's what surprises me more than anything else.
Yeah, but maybe they didn't have enough vaccine.
I don't
It's
know.
possible that.
But you know, in the initially when you don't have a it's interesting to look back at all this. You know, initially when you don't know what's going on, except that it's contagious and it spreads asymptomatically, and you don't have a vaccine, yes, limiting interaction to the extent of even having a lockdown makes sense. In the long run, once you have a vaccine, a lockdown doesn't make any sense unless you're vaccinating people while you're locked down.
Yeah.
Okay.
Yep.
So that to me is a huge sort of weirdness.
well, there's a
I was
lot of
gonna say.
politics. There's a tremendous amount of
Sure.
politics.
So
Unfortunately.
I have two thoughts about it. One, from the standpoint that there's going to be lots of people infected with virus, I find it somewhat worrisome from the evolution of viruses standpoint. Okay. And the second thing is that I think it's now going to be yet sort of another experiment in public health policy, and that we'll learn some
Yeah.
things from that, unfortunately.
Do you think that the efficacy of the vaccine also could play a role in all of this?
I
Absolutely.
think so. Yeah.
That's my understanding.
Well, I was involved in the SARS-1 outbreak because the School of Public Health at Columbia was invited over to China to advise their public health department as to what to do after the initial outbreak had been quelled, and they didn't know if there was going to be a second wave or anything else. So we sat in a room for a week, and we told our stories and we listened to theirs. And in the end, when everybody went home, they fired everybody from the China's public health department, and they reinstalled new people. So that was their solution. They didn't take our word for anything. They just said these people didn't do their job, they're fired. But the next people in didn't know any better either.
I also think the zero COVID policy, wherever there was a case, then they would shut down. I don't think that helped. I think they should have been
It
vaccinated.
just delayed it, right? It just
at the
delayed
same time.
it.
And
That's
now
what
they
it is.
decide. So I read they think now Omicron is mild, therefore they can open up, but it's not because it
No,
still kills a lot of people,
That's-
right?
Oh,
I think if you're immune, it can be mild, but if you're not, it's just as virulent as the other variants. So this is a big problem. And as Kathy said, the evolution is going to be interesting because I presume they're starting with latter-day Omicron variants, right?
Yeah, I was wondering what the variant situation is in China. Does anybody know?
Yeah,
No,
so Daniel said the other day that they have the same recent Omicron derivatives, right?
Okay.
But in the face of such high circulation now, it will be interesting to see if they go further. Because remember, the population isn't terribly immune, doesn't have a high immune.
Right.
So there won't be that pressure initially, just like there wasn't for the first year of the pandemic, right? So be interesting. I mean, it's not a thing you want to see because a lot of people will die, but as Rich said, scientifically, it's going to be interesting. All right, speaking of deaths, unfortunately, we had deaths among the virology community last year. And we had Luc Montagnier, Ovita Fuller, Sam Katz. Did you know that he died? Did you know him, Dixon, Sam Katz? He was at Columbia, wasn't he? Or Duke? I'm thinking of Duke.
No, he wasn't at Columbia, I don't think.
Who's the Katz at Columbia, I'm thinking?
Michael.
Oh, that's Michael Katz, right. This is Sam, sorry. Sam Katz, Neil Nathanson, and Ho Wang Lee. Those are the ones I could find. And no, am I missing any?
I think probably so, but I can't remember.
Yeah.
Okay.
Yeah, let me look at the ASV.
Oh,
Sorry,
ASV
'cause-
has a list, right?
So we also have William Mason,
Bill
Kason
Mason.
King, Walter Eckhart, Mark Collette,
Walter Eckhart?
Ismail Abadia Vargas, and Jim Strauss. Jim Strauss was December 28th, 2021, but yeah, most of those were
Whoa.
2022. Yeah, Walter was
Do you know how old Walter was?
1938 to 2022. You can do the math.
He's 84 years
84?
old. I was born in 1940, so I could do an easy math. I am the elder in that letter.
Elderly.
elderly.
All right.
Bill Mason. Yeah, I remember that.
What is the?
Wow.
Is it asv.org/something?
In memoriam. I don't know. I just know where to go.
All right, I'll put those on from the ASV website. All right, now the other thing we do is we pick our favorite stories/arcs. And since there are 5 of us, we get 2 each. So I will go with my first, which is we had Paul Offit on 3 times to cover this arc of bivalent boosters. So at the beginning of the year, it was decided that we should make a bivalent booster with the ancestral SARS-CoV-2 and then an Omicron variant. And the first TWiV that Paul came on was 9/17. That was in July. And at that point, there had been an FDA advisory committee meeting where the data were presented to the committee, and Paul is on that committee. And that committee voted 19-2 in favor of releasing a bivalent vaccine. Now, remember, at this point, it's already the first variant of Omicron. And then with time, if we get to BA.4/5, and so they make the actual vaccine that's going to be released BA.4/5. And then, of course, by the time it's released, it's beyond BA.4/5. So the first meeting, though, Paul talked about, why he voted no and why the data that he wanted to see weren't there, because at the time they were looking at neutralizing antibody titers that were marginally increased by this bivalent vaccine, and he didn't think it was clinically significant. So then his second visit was 9/42, which was in October, and by then we are releasing these bivalent boosters with now BA.4/5, but the circulating viruses are no longer BA.4/5. And he talked about why they're still being recommended, who would benefit the most for them. But his overarching theme is that he felt that for most people, the ancestral 2 shots and then maybe 1 or 2 boosters of the ancestral was sufficient to prevent severe disease. And he said, we don't have any evidence that the bivalent will do any better. And that's what he wanted. He says it's not really good public health policy to say it won't hurt. Let's use it because it won't hurt. You have to show a benefit. That's always been his theme, show a benefit. He said, most of our vaccines are released because they show a benefit. Now we're moving away from that. And he didn't like that as a vaccine policy. And of course, Paul is a long experienced person in vaccines. And then his most recent visit was the special, where I thought it would be good to make a 30-minute special with Paul, and Rich joined on December 12th, where, again, we had a discussion about the bivalent vaccines. And by then, was it by then? I don't think the CDC had not yet released the data that have been released more recently. But again, we talked about who should get it. He ends up saying, if you are elderly, you could get this booster. It's not clear if it's going to help you, or you could not, and then have a plan like take Paxlovid if you're able to get Paxlovid, because as you know, in many countries you can't even get it. So that's where he ended up. And now, just I think last week, we have 2 morbidity and mortality publications showing early estimates of vaccine effectiveness in preventing emergency department or hospitalizations among immunocompetent adults. And I actually asked Paul what he thought about these. And so there's so first of all, these are people who have received the bivalent vaccine, and we're looking up to 30 days after immunization, right? So theoretically, you still have high antibody and T cell levels. And so they're comparing hospitalization and emergency room visits versus historical numbers using the old vaccine, right? So you don't have a proper control where you have people get a bivalent or they get the regular booster, because you can't get a regular booster anymore. So he thought that was a problem. You can't do historical comparisons of these sorts because different people are getting vaccinated for different reasons. He also thought that using hospitalization was not a good metric, which is something he's always complained about. So we'll put a link to those two. They're two separate studies in different parts of the U.S. And the press, of course, has been saying these studies show that the bivalent boosters work. But I would also say that in a month or two, you're not going to see any prevention as the antibody levels go down and you're back to where you were before. So we'll see. But Paul's final thought is, if you say that these data tell you that the bivalent boosters are working, then you get into a cycle of boosting very frequently. And he says, I don't think we should do that unless we have the data that show that. And I think that's a good point, right? Because the alternative is to boost or not to boost frequently. And so if you're going to boost every year or even 6 months, you should know that that strategy works. And Paul says we don't know from these data whether that works or not. So I thought that was an interesting arc, and we're lucky to have him on 3 times. He likes coming on TWiV.
Yes.
It's a good forum, and our listeners like him as well. Okay. Any other comment?
I'll be interested. I'm glad we've had these discussions.
Yeah.
It's a complicated issue. I'll be interested to see how this all evolves.
And there'll be more data, and we'll talk about it.
One of the interesting things is that collecting the data now is complicated.
Yeah.
Because you can't do a head-to-head trial. All right. So what are you going to use for controls? How are you going to do this? And I'm sure that will evolve as well.
Yep.
And I do always really appreciate hearing what Paul Offit has to say. It's pretty exciting to hear from him.
And I also think, coupled with this is what is a good correlative protection, okay? And that's evolving as well. I've seen some publications recently arguing that, in fact, neutralization titers are probably a more valid measure than some people give them credit for. But that's evolving as well. Running in the back of my mind through this whole discussion is T cells.
Yeah. Well, Daniela Weisskopf will come on and talk about those in January. But there's also been some publications showing that non-neutralizing FC function-mediated activities are important also, right? Mm-hmm. So, and Daniel talked about one of those yesterday. So it can be a combination of everything.
We're learning a lot.
Indeed.
Okay, Dixon, what did you like?
Well, I hate to sound like a copycat, but obviously I love anytime Paul Offit comes on because he's a human being with great knowledge and great tolerance and understanding also for the general public. So when you talk with him, it's not like you're talking to some scientist like Tony Fauci, for instance, and it's not like you're talking to some anchor at a news station either. He tells it like he feels it and knows it. He's very conservative, but at the same time, he can cut you some slack in terms of the way you're thinking about things. So he's used to, compromising at various levels. Of course, if you serve on these committees, I think you have to be. You can't be pig-headed about a lot of this. You've got to be open to other people's ideas. And I just love his style. I would listen to anything he would talk about. I would take a course with him. I would love to know him as a close friend. He appears to be that way. He's very natural, in other words. He's not staid or he's not stuffy at all. And I definitely like that. I think that's an absolute talent. That if you're going to communicate with the public, that's the style. because otherwise they just turn you off. There's another academician, forget about that. Paul Offit is anything but an academician. I know he is, but he doesn't come off that way at all. So I love talking with him, and I love the way his wife interjected herself into a couple of episodes also, and she loves the whole crew. So I think that's wonderful. I mean, it's like a home. It's a home. You get the feeling like you're being, sharing their home with you when he's doing the podcast, and I like that very much. And I think Andy Slavitt, he was a little more rough edged. He was a little more cutting. He had a lot of opinions that were basically his own, but he had a great sense of humor, I think, and that was the one thing I liked about him a lot. He laughed. He laughs at himself. And he laughs at everybody else as well. And he's very iconoclastic, and he's extremely provocative. And I like those two things about people. I do. Not that you can make them all like that, but I think that's the reason why I picked these two guys. And it didn't matter what they said. It just mattered that you could interact with them and express yourself and be appreciated for who you are. So that's why I picked them.
One thing about Offit that Daniel mentioned last night is that he's now kind of been taken on by both sides. So he's a pro-vaccine guy, right? But
right.
Now the anti-vaxxers are quoting him because he's saying, we don't think the boosters are useful. So
I think you're probably doing your job right if everybody hates you.
Well, Tony is the number one person for that job.
That's true. Yeah. Yep.
Yeah, yesterday, by the way, was, or today is Tony's last day.
Last day.
Yeah.
And they still haven't gotten a director for National Institute of Health.
So people are applying, right? Did you apply, Dixon? Or you're not an MD, you can't apply.
No, they rejected me at the door.
Apparently Jeff Taubenberger is one of the applicants.
I think Eric Lander didn't apply either, by the way.
No. He would make it. But Taubenberger would be interesting because he's an MD, PhD, I think, and he has perhaps the right background, but I don't know. He was an interesting guy to talk to. Rich, what do you have for us?
So I liked all of the interviews, and so both of my picks are interviews, and it's really hard to pick, and I certainly don't want to leave anybody out, but I got to pick. And frankly, what just jumps to the top of my list is the interview recently with David Quammen, talking about his book, Breathless. I enjoyed the book. I really enjoyed the interview with him. And my bottom line was, when we came away from that interview, I just found myself thinking, I really like this guy, okay? I want to go visit. I want to hang out. I want to do some coffee.
Yep.
So I thought that was just really a great interview.
I was thinking this morning, so, we've had him and the other guy, what was his name?
Carl
Zimmer,
and then there was one other book writer. But they're all guys, and we need to get some women on this who write. So I'm going to ask
the other guy was
Catherine Wu, right?
idea.
From The Atlantic. She's great.
I will email her. Anybody else I should? Writers, right? I want writers.
I can think of a couple, but I can't dredge their names
There's
up right
another woman
now.
who writes for The Atlantic. Her name escapes me right now. But
okay, let me know, because I think...
There's also a woman named Sonia Shah, but most of her books are more bacteria related.
Yeah, I know that name. I think she did a malaria book also, right?
She has a malaria book, and she's done a lot with cholera.
What about Gina Colada? Has she published anything lately?
I think she still writes, yeah. She would be interesting. Okay, if you think of the names, send them.
TWiV 827.
I always remember her name because it reminds me of a drink I like.
Yeah, the other guy was Greg Zuckerman.
Yeah, that's right. Was that
A Shot to Save the World.
Was that this year? I think so.
That was,
so.
Oh no, it was November 2021.
Okay.
Oh.
Anyway, I want to have a little more balanced view because
I agree.
We can influence people who want to go in the field, and you do that by having the right people who are in it already, right? So, okay, Kathy, what do you have?
So the two picks that I have are about kind of amazing findings in virology that you kind of step back and you go, wow, that seems like a really easy question. And why didn't we already know that? And so I have the quote from Yogi Berra, which is, "You can observe a lot just by watching." And so now I have to digress because Yogi Berra came up a couple weeks ago on a TWiV episode, and I had already gone through this thing about, did Yogi Berra say that or did Niels Bohr say that? The comment about predicting the future. And so I just Googled for Yogi Berra versus Niels Bohr predicting the future. And I got this hit, which is a PubMed paper, which starts out in the abstract. It's by Daniel Dickstein. A quote attributed to many people from the Nobel Prize-winning quantum physicist Niels Bohr to legendary baseball player and philosopher Yogi Berra states, "It is difficult to make predictions, especially about the future." So that solves the question for you as to who said it. Many people said it. Okay. So my first paper about this kind of you can observe a lot just by watching or looking is the TWiV 915 that was entitled Mouse Mouth to Mouse Mom. And the paper was enteric viruses replicate in salivary glands and infect through saliva. It was a Nature paper, a lot of authors, but first author was Suresh Ghosh, and the senior author was Nihal Alton-Bonet. And so I'm not sure how they came to this question. I'm not sure they said, what if we looked in salivary glands for these enteric viruses, or if it was more of an incidental finding or something. But it's known that a lot of viruses transmit or replicate in the salivary gland or transmit by saliva. Well, maybe not a lot. But anyway, we know about rabies and a number of herpes viruses. And this was the finding that enteric viruses do too. And the thought for those viruses has always been that it's fecal-oral transmission. And this finding really kind of adds to that. What they did was orally infected infant mice, and not surprisingly, they found virus in the infant mouse intestines. And they used mouse norovirus and a mouse rotavirus called EDIM. And but they found a spike in secretory IgA in the mouse intestinal tract, and the infant mice don't make secretory IgA, so then they figured it had to be coming from the milk, from the mom, and that led them to figure out that through basically a backwash kind of thing, that these mice that were infected orally, these infants suckling on their moms, were giving their moms the virus infection, and it was replicating in their mammary glands, and then IgA was being produced in the milk. And and they were able to do important things like rule out coprophagy as a source for this. And they confirmed that there was transmission through saliva, and there was replication in salivary glands, in different kinds of immune and epithelial cells. And they also described the use of these salivary gland cell enteroids, I guess, they're called salispheres. And this sort of pointed to using them as a way to propagate some of these viruses that might not be that easily propagated. They were also able to show that human norovirus replicates in salivary cells. And so one of the final things that I think we commented on in the episode was the fact that this salivary transmission, replication and transmission by the saliva might be more important because it could lead to transmission by talking, coughing, sneezing, kissing, rather than the normal way of vomitus and fecal contamination. So those were some of the highlights that I really liked about that paper. But it was just kind of like, oh, well, yeah, it's in the mouth. Maybe it replicates salivary glands.
Yeah.
And it does.
So we got a letter from Nihal afterwards, because we had asked a lot of questions, and she said, yeah, we're working on it. So we hope to hear more about that.
Right.
Brianne, how about you?
So I thought a lot about an arc about spillovers and the many different episodes where we talked about spillover. You can think about whether or not we're talking about receptors or permissivity or things like that. But the two that really stood out to me as helping me sort of crystallize some understanding were first 876, which was Spillover Market with Michael Varobi, where he talked about what were then preprints and have since been published papers describing some of the details of the spillover of SARS-CoV-2 in Wuhan markets. I remember listening to him describe some of the work that they were doing and sort of almost imagined it being in a David Quammen book. I can sort of imagine the book just from listening to him describe the studies and the process. And I felt like that really helped me understand spillover in this case really well. The other one probably is related to the fact that I'm in a biology department. In terms of thinking about spillovers, Vincent and I went to a bat infectious disease meeting in July, and I loved all of that meeting. But particularly the episode of TWiV that we did there, 923, If a Bat Pees in a Forest, Does Anyone Care?, with Vincent Munster and Raina Plowright. And a lot of what they were talking about had to do with ways that ecology and understanding bat ecology or reservoir ecology and land use can influence what we know about spillovers, and how understanding many factors in sort of that ecology side can really help us to potentially prevent or predict spillovers. Raina had talked about given a talk before our episode that I was just in awe of, and that talk is actually those data from that talk are now published in a paper on Nature that is online before print by Ebby et al. Called Pathogen Spillover Driven by Rapid Changes in Bat Ecology. And I have referenced that episode, that paper, the ideas that I learned there, and kind of the new ways I started thinking about spillover from that episode, and really from everything we talked about at that meeting so many times with my biology colleagues and biology students that I know that they are probably some of the things I'm going to remember and take with me the longest out of what we've talked about this year.
Yeah, that was a good meeting. That was a lot of fun.
It was really good.
Another episode that kind of fits, but not completely in your arc, would be the Eddie Holmes one, right?
Yeah, absolutely.
Although he mostly talked about things that aren't true, that are rumored to be true, and so forth. But
yeah,
would fit in.
These two. I mean, I think there are a lot that can fit in. These two, I specifically picked because I felt like they changed how I understood spillover.
All right, so back to me. So the other arc is giant viruses. And, we love giant viruses. And I originally thought, we only did 2 papers. But then as I looked through, I went through every episode. Doesn't take that long. We actually did more. We did at least 3. And, the giant virus meeting last year was canceled, unfortunately, this year, 2022. But it's going to be on in 2023 in Tegger and C in November, so we'll have an episode from there as well. But the first one, I believe, 873, that was back in March. It was called Pig Viruses and Big Viruses. So this is a study on how the mimivirus DNA genome is organized into a nucleocapsid, right? So mimivirus, the first giant virus discovered, has it's not the biggest DNA genome any longer by any means, but it's pretty big, and how it's packaged into that big capsid is interesting. And we did it as a preprint, and now it's been published. But the title was The Giant Mimivirus 1.2 Megabase Genome, 1.2 million bases, is elegantly organized into a 30 nanometer helical protein shield. Let's see if they kept elegant in the title. Yeah, it was published in eLife, and the elegant was retained in the title. It's always interesting to see what the editors take out. And so this is very cool. It's a structural study showing how the DNA is organized into this nucleocapsid made up of protein. It's arranged in 5 or 6 left-handed superhelices that go up and down the protein. And the other cool part is that there seems to be a RNA polymerase subunit stuck on there at intervals. So that would make sense because once it's released, it can undergo transcription right away. So as they say, so it's an elegant supramolecular organization which represents a remarkable evolutionary strategy for packaging and protecting the genome in a state ready for immediate transcription upon unwinding in the cytoplasm. And
I probably brought this up during that episode, but this notion that the polymerase is part of the nucleocapsid is interesting and critical. It is the case also for pox viruses, and there are a couple of papers, actually one that we did, that shows that if you don't package polymerase, you don't make a nucleocapsid. You can still package the DNA, but it doesn't organize itself into a nucleocapsid.
Right. The second episode, 906, back in June, long COVID, giant viruses, and smallish pox. This one, we did virus found in human respiratory samples. This was a paper in Scientific Reports. Detection of virus from respiratory samples in tuberculosis suspected patients. So again, 10,000 respiratory samples were from people suspected to have tuberculosis, and they found 4 positive for virus, 0.4%. And so we don't know what that means. They say further studies are needed because it may be just there, right? Or it may be doing something. It's a small number, but we would like to know if giant viruses are involved in human diseases. So people are looking for it. So you find it there. It's not clear. Viruses infect amoeba. Maybe these people inhaled some amoeba. Who knows? So that was interesting. And then more recently, was it last year? Brienne left. This is bad when Brienne leaves. Oh, no. So let's continue. Anyway, see if she gets back. We just did, I guess, last Friday. Yeah. TWiV 968, a spillover waiting in the wings. We did a preprint, again, bioRxiv preprint on an update on eukaryotic viruses revived from ancient permafrost. So we had a letter from one of the authors, Jean-Michel Claverie, saying, people say, it's rare to isolate viruses from permafrost, which they had done a number of years ago, 30,000-year-old ice core from Siberia. And he said, no, it's actually not so rare. We have other samples, and I'm just clearing out my drawer and publishing them in this paper. So they showed a number of new infectious viruses because they use amoeba to try and infect with melted ice or intestinal contents. And they find 13 new viruses isolated from 7 different ancient Siberian permafrost samples, the oldest of which is 45,000 years. So it's very interesting because there's this idea of as the ice melts, are we going to release viruses that could be dangerous to us? And that's something we talked about quite extensively on that episode. So those are giant virus episodes. I really like giant viruses because why do I like them? Well, her computer froze working on it. Okay, good. Why do I like giant viruses? Because they're so big and that presents unique problems to be solved. And, one of which is packaging the genome, as we saw, but also they have a lot of genes that are novel. We don't even know what they do. And we don't know what hosts they infect. They don't all just infect amoeba, I'm sure. That's because we're using that as a sentinel, but I think they're quite interesting. Dixon, what do you have for us?
Well, it's back to another interview that we had with Dan Wilson, getting the, debunking the funk.
Yep, that's right.
And I just found him charming. He was, for such a young person, to have such insight into the fact that he was at one time on the opposite end of most of the arguments that he now champions
Yeah, that was interesting.
tells me
yeah.
that, anybody can do this. Damn it, anybody can do this. They just don't want to. And he obviously wanted to. And it was a pleasure to talk with someone at another level. Again, I'm using the Paul Offit reminiscence as a way of saying, here's another person that talked straight talk, didn't pull punches, didn't use jargon, and said, well, let's follow the data and see where it goes. And most people don't know what data is. They obviously don't. They think they're a collection of facts, and actually that's not true at all. The data are the data. I mean, nobody owns it, so everybody can use it any way they wish. And it's too bad that a large organization like NASA and NOAA have accumulated huge amounts of data, which people cherry pick, and they say, oh, there's no global warming. Look, the temperature is dropping, the ice sheet's increasing. Yeah, that's data. But if you look long enough, you're going to see that the opposite is actually true, but that doesn't support their argument. And I thought he was wonderful at getting to the essence of the deniers and then supplying them with unanswerable questions unless they opened their mind. And that's the whole secret to good biomedical research, is just go with the data. Go with the data. Rich, you keep saying that, and I respect you deeply for that. And I think all of us on this team all did that while we were conducting our own research.
Dixon, you were saying you were finishing with Dan Wilson, right?
Yeah, I think I just summarized by saying
I wholeheartely agree, Dixon. This was, as I said, it was hard for me to pick amongst the several interviews that I liked, and this was one of them. I think Wilson is actually a remarkable guy. I think it's worth watching him do what he does to learn how to process data, to learn what critical thinking is.
Right.
If you just want a lesson by example, just watch this guy. He's very good at it.
Yes.
And what I like is that, so we started TWiV. We had no intention to do that kind of thing. We just wanted to talk about the science, right? And people say, oh, you've captured this virus podcast arena, but there's always more. And he found a little niche, which is to take things people say and tell you why that's wrong. And it's not what we want to do particularly, right? Because we can't do everything. We can't even get to all the data. We have a lot of guests, and we barely do papers anymore. Hi, Brianne.
Hello.
Hey, she's back.
She's genuinely back. That's good.
Yay.
By the way, you say we barely do papers anymore. I find that over the 10-year history of this, I now read papers completely different, not completely, but quite differently than I used to before.
How so?
I've gotten much better at sort of understanding what's going on a lot quicker. All right? And I have to say that knowing that Vincent is going to lead the discussion,
Here.
I lean on that a lot. I would read them quite differently if I knew I were going to read the discussion. I know how much detail I have to assimilate in order to be able to be a color guy as we go along. Okay, so
Bless you.
Thank you.
Can I hear your voice? I want to see if it's loud enough.
Hello.
Okay. The mic is working, right?
Yes, the mic is working.
All right, good.
Good deal.
It's interesting. In the other podcasts, I don't lead everything, right? Because they're not my expertise.
Right.
We take turns typically, but I do take turns even on TWIM and Immune and TWIP and Twin even. I can occasionally, I'll find a paper with a virus that gets into the CNS. I can do that. So it's interesting that it has evolved this way. And I think you guys are okay with that, right?
yeah,
Absolutely.
Yeah.
No, I sit here in awe.
But I think
really,
that you guys make great commentary. That's what makes it work, right? I'm kind of feeding the tape through, and you guys. So I think it all
Vincent,
works.
We were colleagues for many years, and I
Aren't we
knew
still?
You. Yeah, no, of course. Well, I'm retired. Who knows what? I mean, when you just say hello to somebody at a faculty meeting and sit next to them and share a sandwich, and you go back to your lab, that's the end of that one. But to have the privilege and pleasure of spending so much time working our way through how do we do this, here we are, 14 years later, contemplating year number 15, with 1,000 TWiVs.
Amazing.
That's absolutely amazing.
No,
you know,
it's true.
even the Marx Brothers didn't last that long. Did you hear why they broke up, by the way? Because they just featured Groucho on the Dick Cavett interview on PBS. It was wonderful to watch. They were making a movie. The Marx Brothers were making a movie that they never finished. And I believe it was Harpo, but it could have been any of the others, turned to the other Marx Brothers and said, "Are you guys having any more fun?" And they looked at each other and said, "No." And they just got up and walked off. And that was the end of that. The movie never got made. They quit. They went their separate ways. And they said, "Okay, it's gone." And I can imagine the Beatles ending like that. And the Rolling Stones have held together pretty well. But for the most part, even close-knit groups like Bob and Ray and lots of other comedy teams, Dean Martin and Jerry Lewis is a good example also. When their personalities began to change, they became incompatible. But that hasn't happened here.
I often wonder, I wake up one day and someone says, I've had enough of this. Really, I don't want it to happen. I mean, I don't know any of the podcasts, but we've been remarkably lucky. We've asked people to leave, but no one has left.
No, Vincent, I'm not saying it's been perfect either. It has not been
of course,
perfect.
nothing is perfect, of course.
But it's
we have evolved. We have changed a lot over the course of
There is a glue that
years.
holds us together, which is
I'm fortunate that you guys all do this, that everyone else does it. I couldn't do it on my own, and I appreciate it. But as I do worry that one by one, you guys are going to say, "I've had enough of this," and it'll be hard because it changes. You know, even if you're not in every episode, when you're here, it's an entity, and but that's life. You have to deal with what people have to do. So I think we're lucky to go so many years and basically have the same crew.
Agree.
Right?
I totally agree.
But Dan got a nice TWiV bump on his YouTube channel. A lot of people discovered him as a consequence. So that's really good. That's really good.
Excellent.
All right, back to
We'd
Rich.
like to have him back.
Sure. Rich.
Another interview. Mershlinsky versus monkeypox, or at the time it was monkeypox, now it's mpox.
Right.
I want to reiterate that this idea came from Ed Niles to interview Mike. And it was more or less at the peak of the monkeypox epidemic. And I think there was significant confusion about what was going on and some misinformation as well. And if anybody should know monkeypox or mpox in all directions, in not only the basic science, but also from the point of view of the FDA dealing with vaccines and drugs, and BARDA dealing with how to assist with the public health challenge. It's Mike. And he did a great job, and I think set a lot of people straight. And so, as a matter of fact, you may still be confused about monkeypox. Go back and listen to that episode. It was really very good.
Yeah, I think it was great at explaining all the things that people didn't understand. He was really good about that.
And I actually just looked up. There's a CDC site that is U.S. case trends for monkeypox. It's an epi curve, and it's down to very, very few.
Yeah.
5 reported on December 21st, as opposed to a peak when we were up near 500. So That is essentially over.
Yeah, it was a good, although initially it looked like another public health failure, right? Daniel often called it that, but we did come around. It was good. And I think obviously having those vaccines there, right, Rich? Boy, that made all the difference.
Oh, yeah. Vaccines. And I'll be interested in a retrospective of how big a role the drug played
Yeah.
in this.
Yep.
TPOXX or whatever.
Tecovirimat, right?
Tecovirimat, whatever we're calling
Reminds
it.
me of
now.
an automat. You know, when I was a kid, my parents used to take me to the automat here in New York City. That was a big, exciting thing to come.
Oh,
You put
yeah.
do you remember Dixon?
No,
You
but
would
Marlene
put-
does. So I didn't. I grew up in Jersey, so I didn't get the privilege of.
Yeah, we would come in for a visit, usually
No,
around
we
the
never
holiday.
went to the automat.
And you
Never
put
went
your
to the
quarter in, you get something to eat out of those little boxes. That was so unusual. The automat. There's a cool version of that in Japan. You go to a restaurant and they have these conveyor belts going
Oh, yeah.
past
those
your
are good.
table, and you just pick off the plates
We
that
have
you
that
want, and
Yeah,
then they
that's
charge
right.
you per plate.
Exactly.
So cool. All kinds
Yeah.
of sushi. We have that here, really?
We
Yeah,
have one
there's a
called
couple
Choo
of sushi
Choo
places like that.
Sushi,
Okay.
and there's a model train that goes by, and you pick your food off. Of
Yeah, Choo Choo Sushi. That's wonderful.
that. That's good.
Kathy, what's up?
I forgot to mention with that first paper that Trudy Ray wrote a blog post about it, and that was, where do I have it? August 4th in the Virology blog that Vincent puts out. And then this paper that I've picked for my second one, I already know is going to be a Trudy Ray blog post, so watch for that as well. And this is another one in the theme of sort of simple questions. You can observe a lot just by watching, and it kind of answers the simple question of, well, what happens if you infect a cell with two different viruses? And it's a Nature Microbiology paper talking about hybrid virus particles that are produced by co-infection of influenza virus and respiratory syncytial virus, viruses that are now more familiar, or at least RSV more familiar than it had been in the past to a lot of people. And it's a group from University of Glasgow, and the first author is Joanne Haney, and the senior author is Pablo Murcia. And basically, people have done experiments where they've infected with different viruses in the past, and generally you get what are called pseudoviruses, where it's predominantly one virus and it incorporates some of the envelope proteins and therefore has some appearance like the second virus. But with this, they found pseudoviruses, but they also found true hybrid viruses. And so what they used here at first was a human lung cell line, and then later in the paper they used primary human lung cells. But in both cases, it's all in vitro. So this isn't talking about what happens when you co-infect two animals or co-infect two people. One animal with two viruses or one person with two viruses. But it is all in vitro, so it has that caveat. But the important thing was that they found these hybrid viruses, and the EMs and the various images that they have of these are just phenomenal. So they did high multiplicity infections so that they had a good chance of getting both viruses into the cells. And then they used all different kinds of microscopy, and the pictures are worth 1,000 words. I know Vincent used the image for the show image from that TWiV, which was 958, and it's worth going to look at that for sure. And so these hybrid particles had not only the glycoproteins, but they had the RNPs. And so what that means is that they had the genomes and the genomes associated with the proteins. And so that is what's different about these viruses than just pseudoviruses. And it's interesting because the two viruses, of course, are both respiratory viruses. So you could imagine that you might know that you're infected with one, but not realize you're infected with the other. And they're both negative strand envelope viruses, but one replicates in the nucleus, that's influenza, one replicates in the cytoplasm, and yet they can make this hybrid kind of monster that comes out of it. So everything's not equal. There are some aspects of it where the respiratory syncytial virus has a little bit more of an edge than the influenza virus, but they show that I've lost my train of thought here. That they look at the receptor tropism, they look at neutralization, and they look at whether or not these things are infectious, and they are. And the significance is that you could imagine that you might expand the tropism of one virus or the other by having it sort of cloaked with the proteins of the other, but yet have both genomes there. And it could somehow maybe enable immune evasion. So to me, it was just a really cool question. And although people had looked, they hadn't looked in this way before. And then they had this fantastic microscopy that added to it.
It's a great paper.
Yeah.
Another one of those, wow.
Yeah.
Is there any comment in this on how frequently or the likelihood that in nature this happens, that an animal has cells that are co-infected with two different viruses? I've wondered about this.
Yeah, I don't remember seeing anything like that.
They do.
So.
I think they say they do some speculation, if I remember, saying that the multiplicity is probably high enough to get co-infections. And they were going to look to see.
Right.
Yeah, but you'd have to imagine the timing would be really important too.
Yeah, of course.
You wouldn't want the one cell to start going into an antiviral state before the second virus came in.
That's right. Mm-hmm. So this, I went to the video to see how many downloads. This is a typical one, almost 7,000 downloads on this video. But I also noticed, for some reason, if you scrub the little control to move the video, it comes up with at different parts what people are saying, and right at the beginning it says Dixon, Dixon, D Palmieri, Depalmieri. They made him into an Italian, Di Palmieri.
Thank God they won't know who that was then, will they?
And then if you scroll along, you see deer behavior, zoonosis, study results. But in the very beginning, it says Ian Arbor, IAN Arbor.
Oh.
It's so funny how the bot does it. Oh my gosh.
Yeah.
Ian Arbor. Kathy Spindler from Ian Arbor. Anyway, that's great. And that brings us to Brianne.
So when I looked through the list of episodes for this year, there were a lot that I could have chosen. But the reason why I chose this one is actually that even before I started, I said, man, that was one that I really remember, that EBV episode. Was that this year? If it was this year, I'm definitely picking it. And so I was very excited to see that I had remembered it, and it was, in fact, this year. This is from February 2022. TWiV 869, Epstein-Barr virus, and MS, a perfect storm. There had been a number of episodes before this where some hints of herpes virus infections and different types of neurologic diseases were sort of out there, and they were always little hints. And in this episode, we talked about two papers, one of which still kind of floors me with the epidemiological work that they've done. And so this paper was a paper that was in Science in the sort of beginning of January last year, called Longitudinal Analysis Reveals High Prevalence of Epstein-Barr Virus Associated with Multiple Sclerosis. This used data from U.S. military recruits over 20 years, such that they could look at 10 million subjects and see 955 who had MS during their time in the service, and actually look at Epstein-Barr virus infections and how that was related to MS. And so using these huge numbers of subjects, a statistical association between getting MS at a particular part of your life getting Epstein-Barr and proceeding to MS. And so I just thought that this was really amazing use of data to start to study something that we had a lot of hints about before, And then in that same episode, we talked about a Nature paper that looked at antibodies in MS patients that bound to some EBV proteins. And so these were both two really nice papers that kind of moved forward this idea of EBV MS. I thought that the epidemiological paper kind of helped me understand some issues of immune tolerance and ways that one could see tolerance be broken. And I think it's one that will probably have a lot of future study going forward.
I bet you also looked at the IL-1A paper, right?
That was my honorable mention paper. You're right.
Yeah, that was you mentioned that on immune, though.
I mentioned that on immune,
Yeah.
and it is also one of those papers that I think back on very frequently.
It's very cool. Yeah.
And that I am designing experiments to think about often.
But these associations are very difficult, but these two papers really show how you can do it right and get some confidence that actually this is what's happening. Yeah.
Exactly. You know, I can't imagine looking at as many records or as many samples as they did. And I'm so happy that they did to give us these insights.
Yep. All right. That's our little ARC review. And now another thing we started not too long ago, favorite episode titles. So my favorite was TWiV 920, Science Will Break Your Heart, which is something that our guest Andy said. He was going at the end about how you have to really love it because at some point it's going to break your heart. And he was totally right. And I love it. And so this is, I think somebody made a T-shirt on our Cafe Press site of this. So that's my favorite.
Yeah, that was at the ASV meeting. So
Right.
Andy Maley.
Dixon, what about yours?
I had to go with From Lizards to Lhasa with Tom Monath. Tom and I became friends a long time ago when I was an active member of the ASTM&H Council. And I always gravitated towards his inner circle whenever the meeting was held, no matter where it was held. And I being the fly on the wall, as I am here, a talkative fly, but nonetheless, to listen to all the wisdom and all the humor and all the pathos that a group of people that have a common goal, pursuing an issue that relates very much to the public's health, and to see the various approaches that each one chose. But Tom is he's as in control of the moment. That's all I can say. Tom is in control of the moment, and he never, ever, ever steps out of that mode. I've known him for many, many, many years now, and the interview was as typical as it was just sitting down and talking with him, as if we had just had lunch last week. So I chose that one from my heart rather than my head, but it's a great interview anyway.
Thanks, Dixon, because that one was also way up on my list of favorite interviews.
Oh, good.
Tom is a remarkable person.
I totally agree.
What about you, Rich?
For no reason other than just the title itself. If a bat pees in a forest, does anyone care? Great. I guess the answer is yes.
My understanding from Rena was if it pees in a forest and there's nobody around, the answer is no.
She said that. Isn't that her mention?
Yeah. It was.
Yeah. Kathy.
I chose Driven to Immunodistraction.
That's good.
So it tells you, of course, that Alan was on the episode because I'm sure he helped figure out that title. And it was something about immunodistraction. And I'm now looking at the summary that Vincent writes for every episode. And I was clearly distracted enough that it didn't sink in or last till now. But that was TWiV 931. You can go look at that.
Yeah, it's about antibody responses to influenza virus. It's very cool.
Right.
It's a good title. Yeah. Mm-hmm. And Brianne. Yeah,
I chose TWiV 887, Bonfire of the Monocytes.
It's good.
I really like the paper that was discussed. I really like the science behind it. And I just think that's a cool title, too.
Right.
It is.
And Alan, it's got to be an Alan title. He actually wasn't on the episode, but he tends to sort of sneak in the back door and look at the Google Doc and drop a couple of titles in his spare time.
You are right.
I want to thank some people who have helped us throughout the year, besides all of you, of course. The timestamps you see on every episode in the show notes are done by Jolene Ramsey, who we met many years ago visiting Texas A&M. And in fact, that's where these pillows come from. Her mom made those.
Also, you and I met her at Indiana.
Before that, that's right. Yeah.
Right.
And she also maintains a page called the TWiVerse, microbe.tv/TWiVerse, where we put poems that have been submitted by our listeners. And of course, I don't think there have been recent ones. Although there was one submitted to, there's one
Last
submitted
week?
to Daniel, by a physician about Paxlovid. That was good. That needs to go up there, Jolene.
Mm-hmm.
And I also want to thank Hannah Berhe. Hannah is a master's student here in New York at NYU, and she is an intern for Parasites Without Borders, and she helps us with the show notes for Daniel's TWiV clinical update, because they're quite extensive. So I am very grateful that she does that, because it's a lot of work. I also want to thank David Ramada, who does the video editing for Daniel's TWiV, just the video. Hopefully he will do more for us. I call on him from time to time to do video edits when I don't have a chance. And we also have a new audio editor, Peter Duff, who's from Georgia. And he just cold emailed me. He said, I'm a professional audio editor. And so I said, help me out. And so he edits something every week. Usually it's not TWiV, because we record on Friday and he doesn't want to work on the weekend. But if something else comes up during the week, he did, for example, the Carl Zimmer, the Taubenberger, a lot of those other episodes. So if you see a slight change in the sound, it's because of his touch. He has a different touch, of course, than I do. But I'm happy to offload some of that. My goal is to eventually offload all of it so I can focus on content creation, as they say. And Kara Pound also joins the team. Kara's from Florida and originally was working with Parasites Without Borders. And I decided to bring her on as a publicist. She's going to write
She's good at that.
a monthly newsletter for us, where there'll be a button you can push to donate. And she said that really helps with every time she sends that out for Daniel, you get a little bit of money trickling in. So you're leaving that on the table. She's going to help take care of social media for me. She'll have access to all the platforms so she can do things when I'm not there, and other things as well. She'll be helping with. Really good. I'm looking forward to getting more of that. And then, of course, there's the incubator here where I am in New York City. It's on 7th Avenue, 2 blocks from Penn Station. You're all welcome to visit. Not all at once, but one at a time. We had a relatively well-known actor who was going to come last week, but he decided there was too much COVID. He lives in Brooklyn, and he's been on Sopranos, Boardwalk Empire, Orange is the New Black. Okay. And I thought it would be cool to have a picture with him and have him do a little video clip and say, I love TWiV or something, you know. But he was going to bring me Pignoli cookies from Brooklyn, actually. So you got to bring them at some point. So he said he would come next year. But the incubator is our recording studio. It's pretty much done. There are 2 parts. They're here where I'm recording. On the other side is just an area with a table, some chairs, a little library, a kitchen, and so forth. And we hope to host number 1,000 here or maybe somewhere else. I'm not sure. We have 116,000 subscribers on YouTube. We just hit that. That's great. I mean, my goal is right now 200,000, and then we'll go for 1 million. And I want to thank everyone who has contributed financially to support us through Patreon, PayPal, checks sent through the mail, get a lot of those, or the Amazon portal for purchases, Amazon Smile, you can do that. Parasites Without Borders fundraiser, which is still ongoing over at parasiteswithoutborders.com, and vaccinated.us, buying spike shirts, which you won't be able to do when you hear this any longer, but that ended at 2022. We got a very nice amount of money from Matt over at vaccinated.us. So all of you who have contributed, thank you so much. We are a tax-free entity, a nonprofit, so your contributions are U.S. federal tax deductible. And if you want to contribute next year, please go to microbe.tv/contribute. We depend on your help. We don't take ads. We don't monetize on YouTube. We depend on your charity to keep us going, and we really appreciate it. We do need it. As you heard, I'm employing more people to help me out. My goal is to have even more people. We have some ongoing expenses like internet and so forth, rent, and we have pretty much all the equipment we need. But recording suites like the one we use have a monthly subscription, so we have to pay for those as well. So we thank you all for your support, for helping us to disseminate accurate science information. I would like to do one round of emails, but first I want to show some Christmas cards that we've received or things we've received here at the incubator. Here's one Christmas card which came in. It is from Jan T. and Joel. TWiV, happy holidays with utmost appreciation for you all. So it's a cool one that opens up and becomes 3D.
Oh, pop-up.
So thank you, a pop-up. And then we have here a dog on a card with a very nice handwritten note. And it says, "Dear all you all, at
All y'all
at TWiV and etc. Thanks for the words of sanity in this totally crazy world. Cheers, Deirdre." So thank you. That came with a check. Thank you, Deirdre, very much.
Lovely, lovely.
And then this one also came with a check from Susie. I don't know if you can see that, but Susie writes, "Vincent and crew, thanks for the stickers. And again, thanks for all that all of you and your guests do. Susie is a school bus driver in Tennessee, which is great, right? Some of the,
Yeah.
I mean, we want to reach everyone, but when we reach people like this, I'm really pleased, especially. And so let's do just the round of email. We have actually a poem. So Dixon, you're a good poem reader. Why don't you read that first one? I don't think so.
I would be happy to. Eugenia writes, gaslighting. That's the name of this poem. Merriam-Webster Word of the Year. We live in a world of lies, misunderstandings, and deceptions, making us call into question our firmly held conceptions. Our circular thoughts seek truth, but there are few straight replies. That's what goes on in a world of misunderstanding and lies.
Very good.
I like it. I hope that wasn't computer generated.
I don't think it was, no.
No, it sounds fine. It sounds
That
fine.
was
So-
Eugenia generated.
Yeah,
I,
that's right.
Eugenia generated.
So I asked earlier this week on Twitter, why don't you send in your favorite or least favorite episodes or anything? And 2 people responded. So first one is from Liz, who writes, favorite episodes, all of them. I can't pick one. Love Dr. Daniel Griffin's quote of the week. Never stop providing a quote. Pick of the week, Professor Racaniello's recommendation of author Andrea Camilleri. Rich's book, Mindfulness in Plain English. Dixon's recommendation for jazz. So she has favorite picks of the week. Sorry, I can't narrow my answer. Liz is in Columbus, Ohio. And Liz wants to come to ASV when we're there. I said, sure, no problem. Just find me and I'll get you in. Kathy, can you take the next one?
Mark writes, Dear Vincent and other TWiV hosts, congratulations to you all on another year of TWiV. 2022 was an excellent vintage year. Year-end is commonly a time of pondering the year past and preparing for the year ahead. Below are opinions from the longtime listener of the best and worst of TWiV in 2022. Best episode, TWiV 876, Spillover Market with Michael Warraby. Why? Warraby tracks evolution of SARS-CoV-2 spread in Wuhan. He developed dissemination models and tested them against spread of 2 variants in Wuhan. His models matched reality. Warby presents facts and logic that lead to the conclusion that COVID-19 spread from a wet market. Applying Occam's razor that the simplest explanation for an event is to be selected over alternate, more complex ones, he concludes zoonotic transmission from one of several possible hosts in the market was the source of the virus. Doubters will doubt, haters will hate. Conspiracy believers, let's call them conspiracialists, will suspend logical thinking and clutch at their favorite explanations for COVID-19's origin. Lab leak, created by the CCP, or some favorite explanations. Sadly, the proof offered by the now he's changed it to I just didn't read it right. Conspiracialists is a repeated recitation of conclusions without logical support, shouting, or the ultimate proof, Fauci did it. Best story arcs. During 2022, as in past years, some topics, ideas, and stories repeated themselves in small bites. I think the best were 1. Bats and other hosts. Frequent discussion on how SARS-CoV-2 infected different animals and what the implications are for human health, or for understanding the origin of this virus. 2. Deca picks. I'm creating a new conjunction here to describe Dixon's 10-part listener picks for comedians and jazz musicians. 3. ABC, anything but COVID. It was refreshing to see other virology stories and topics emerge that were not chained to COVID-19. Your multiple guests and episodes recorded from travel conferences were wonderful and refreshing alternatives to incubator-based studio discussions. Moving on. Worst episode. TWiV 929, Empathiology with Andy Slavitt. Why? Slavitt didn't offer any clinical or scientific insight. He came across as a smart aleck boasting about his past achievement in the Obama administration and his work in the Biden administration. He denied, jibed, and threw up shrapnel whenever the conversation came close to being critical of the Biden administration's missteps in dealing with this health problem. It's a joyous time of year. I am sure I speak for many other TWiV listeners in hoping that Andy didn't injure his arm or hand from patting himself or congratulating himself for the job he did. Moving on, here's my last listener pick of the week for 2020. It's George Carlin speaking at the National Press Club, making fun of the murky, vapid, and hollow language politicians use. Would you be surprised to know that he doesn't use any expletives in this segment? Watch here.
Yeah, I would be surprised.
And he provides the YouTube link. Looking forward to a great 2023. Mark. P.S. It's very rainy here in the Bay Area as I write this. PPS, be nice to Dixon.
I think I've been nice to you for this year, right?
Extreme. Everyone has been remarkably nice.
Because the previous year I wasn't nice all the time, but I've calmed down this year.
Water over the dam, kiddo.
Thank you, Mark. Mark is a long-term listener. Mark used to work for Drobo, and years ago we gave away Drobos, and he was responsible for that.
Yeah.
I was at an ASV, Kathy, I think it was Minnesota, right, Minneapolis. And someone ran up to me and said, I got one of those Drobos.
Wow.
That's very cool.
Cool.
Brianne, can you take the next one?
Sure. Laura writes, Dear Dr. Racaniello, I just wanted to say how much I appreciate the comments and wisdom of Dr. Depommier on TWiV. It is enlightening to hear him incorporate his general knowledge of biology and ecology into the virology discussions. Your program is so much richer because of his insights. Kind regards, Laura.
There you go, Dixon.
Here. Yes.
Oh, shucks.
I thought you would like that.
Yeah.
Gift to you. Why don't you take the next one too, Brianne?
Sure.
Right.
Lisa writes, there is a session going on about the mortality rate of those of us who have had the COVID vaccine and boosters right now from Wisconsin. There are virologists there saying that the vaccine is basically going to kill me earlier than I would perhaps die if I hadn't had all the pokes. I am freaking out. How in the world do I find out what is real and true? It is being streamed through the Epoch Times, and I have read that there is evidence of misinformation coming from this group. I am a TWiV listener now, but was not back when we were in the throes of COVID. Would you recommend a certain episode that I go back and listen? Thanks, Lisa. I think we have talked about Epoch Times before, and from what I have seen, this is completely untrue. There was a study that came out somewhere in Scandinavia looking at some early vaccine recipients and what fraction of them died after receiving the vaccine. And if I remember correctly, they were all the elderly. And there was no control of just how many members of that same age group passed away who hadn't been getting the vaccine. Those tend to be the types of data people are using when they talk about this mortality rate with the vaccine. But I don't have a specific episode to point you to.
You will not be killed by the vaccine.
No.
You will not be killed by the vaccine.
Wikipedia says that the Epoch Media Group's news sites and YouTube channels have spread misinformation and conspiracy theories such as QAnon and anti-vaccine misinformation and false claims of fraud in the 2020 United States presidential election. That's just a one-liner from an extensive wiki article that backs up those claims and gives some insight into what this is about. Do not pay any attention to the Epoch Times or anybody who refers to it authoritatively.
I really think that in the very beginning of COVID, we spent some time talking about the Epoch Times and the folks behind them.
We certainly reviewed the vaccines in multiple episodes last year, I would think, right? Let's see, TWiV 683, 2 COVID-19 mRNA vaccines. And I'm sure we looked at the safety data in that episode. It was November 2020. So just, how in the world do I find out what is real and true? If you want to find out about viruses, this is one good place. Dan Wilson's Debunk the Funk, there probably is an episode about debunking anti-vaccine sentiment with respect to these vaccines as well. But don't listen to the Epoch Times. Absolutely not. It is a font of misinformation, for sure. Okay, one more. Rich, you haven't read, correct?
Correct.
All right, take the next one, please.
David writes, Dear Vincent et al., I'm listening to TWiV 960 and your wonderful conversation with Dan Wilson. When you get to about 87 minutes, Rich initiates a discussion about being wrong and its value for learning. I wholeheartedly agree. However, there are two kinds of wrong. The good wrong is hypotheses that we develop from our data that help us design good experiments and disprove them, as Dixon says. The bad wrong is confusing an hypothesis for a conclusion. A potential antidote to the second kind of wrong actually surfaced a few minutes earlier when Dan used the phrase, not supported by the data. Being careful about how we frame our conclusions should always be at the front of our thinking. And you talk about it all the time. When we do outreach, we need to train ourselves to be hyper careful to communicate in terms of what is supported or not by the data, and clearly separate that conclusion from hypotheses. That's a key part of educating the press and the public as to how science works and how they can best incorporate scientific findings into their thinking. It's a long, hard slog to change the old mindset, and there's always the possibility that they may not like it because it's not sexy enough and they stop using you as a source. And maybe more attention to distinguishing between wrong that helps us learn and the wrong that doesn't will make us better at what we do. Of course, none of this is new to you as the individual pieces come up repeatedly in your podcast, but I thought it would be useful to make an attempt to pull it all together. What can I say that hasn't already been said about the work you're doing? Huzzah, Dave. And Dave, this is, I think I can say, Dave Spector, emeritus professor from Penn State Hershey. Thank you, Dave.
Yeah.
This reminds me,
good.
I think yes, this is excellent. I want to tell a little anecdote here that I think I probably told somewhere in the past. What did we decide? 13 years?
14.
I gave a graduation talk once when I was still employed to a small class of graduates from the graduate programs in the Health Science Center, where at some point in the talk, and the audience was a mixture of graduates and their families and friends, okay? At some point in the talk, it became appropriate for me to outline the scientific method, which I described as: you formulate a hypothesis, you design experiments to test the hypothesis, you examine the data to see whether or not it supports the hypothesis, and if it does not, then you change the hypothesis and go through and reiterate the process. And when I said that if the data don't support the hypothesis, you change the hypothesis, there was an outburst of laughter in the audience.
Right.
And that was totally unexpected. And then I thought about for a long time afterwards, and I realized that it's quite possible that the notion that a hypothesis is, not necessarily something that's going to be true or immutable is a foreign concept. That people
Right.
are used to the notion that if you've got an idea about what might be right and you find that the data don't support that, then there must be something wrong with the data. Okay?
That's right.
That you must be right. Okay. And
That's
no,
right.
no. A hypothesis is something. Yeah, you may firmly believe in it. Okay, but it's there to help you construct the experiments to test
Sure.
it. Okay, and if the
Yep.
experiments don't support it, then you got to change it. Weird.
Right. That's right.
Yeah. Yep.
Bingo.
Nope.
But it's a start. It's a start.
Yeah, absolutely.
You know
I
what?
remember-
It's the beginning. It's the start. It's not the end. An hypothesis is the beginning, not the end.
Yeah.
I remember putting that in grant proposals and saying, and if we get this data, then it will not support the hypothesis, and we will,
Right.
design another
Right.
hypothesis.
Right.
And I remember giving it to someone to read at some point, and they said, "You can't say that." I was like,
Exactly.
"Watch me."
Yeah, exactly.
Funny.
Yeah. Wow.
I meant to mention the first letter writer, Liz. She sent me a gift along with
right.
some support for Microbe TV, which we thank you for. But this is cool. It came in this cool sack, and this is a pair of socks with guitars on it. Very cool.
Oh, cool.
Thank you, Liz. And it's because in my home studio, there's a guitar in the background, which hasn't been used in many years. Okay, time for some picks, our last picks of 2022. Wow, we have a lot of letters here. Look at this. This is very cool. We'll have to get to those next year. Dixon, what do we have today?
Well, we're continuing on with the Jazz Project, as was alluded to in some of the letters that we just got. We're up to the tenor saxophone section of the the large jazz band, but tenor saxophone players that are outstanding and have set the stage for everybody else. John Coltrane, Dexter Gordon, and Stan Getz. Now, there are many, many, many other names that could go here too, but these are the three that have, if I sat down and that music came on, I would never turn it off. I would just keep listening. They're remarkable players. Every one of them is different styles, different phraseologies. They take the same instrument and they get three different hypotheses out of it, which you can either accept or not, and you can continue on down the road. John Coltrane, his signature album is Giant Steps, and a lot of you might be familiar with that already. And his signature song is Blue Train. There's nothing more lyrical and deep in terms of musicology than this one song, and everybody will recognize it when they hear it. I'm sure it's been played many, many places and many times. Dexter Gordon, who a lot of people are not familiar with, but they're familiar with the fact that he was the star of the movie called Round Midnight, and he actually played himself in that movie. And it's a wonderful movie that you should see because there are hints in there as to how to listen to jazz and it involves a sax player and a woman vocalist who's trying to make her way through the nightclubs of Paris. And he advises her, listen to the bass. Just listen to the bass. Don't worry about the piano. Don't worry about the guitar. Don't worry about the saxophone. Listen to the bass. And that's how you should base your music. That was a remarkable insight. I would have never, ever guessed that musicians have, I guess, I don't know, a signaling pattern which emanates from the bass and goes out. And that was a remarkable insight. Stan Getz, you might know his music recently, but he started with Charlie Bird, who was a guitar player, a remarkable guitar player, and they both teamed up for their album Jazz Samba, and the signature song Desafinado. You would never forget it if you heard it. It's gentle, beautiful, rhythmic, lush. It's Saturday. I call it Sunday morning breakfast music. You wake up, you've got nothing else to do. You turn on the radio. There's some Brazilian samba music. You've got some eggs cooking away. The coffee smells. The bacon is going. Your wife is not yet awake, or your husband is not yet awake, and you can have this brief moment of time when you're just enjoying a perfect half hour. And this music is a perfect half hour, I would say.
Very nicely put, Dixon. This is number what in the series?
I'm not keeping track of numbering, but
there's more. There's more.
Oh,
correct?
there is. Oh, yes, there is.
Great.
I have to conclude with the GOAT.
Yeah, we're not getting to that yet. Okay.
The greatest of all time. Yeah, and I haven't even touched on that yet.
Okay.
That's right.
Thank you, Dixon.
You're welcome.
I see that Rich put in a link.
Yes.
Yeah, there's a Richard Condit who's a saxophonist who's on the music faculty in Lamar University in Beaumont, Texas.
Oh
yeah,
my goodness!
I got an email from him.
How cool!
Because he'd been invited to a scientific conference in Dubai, and he looked for other Richard Condits who might be scientists around. The irony is that it's not me; it's another Richard Condit who's a tropical ecologist that gets confused with me all the time. So now we have a three-way conversation going on. We've agreed that we all three of us ought to go to Dubai. But I want to meet this guy sometime and listen to his saxophone.
That's
great.
Cool.
Oh, yeah.
Cool.
Yeah, absolutely.
Brianne, what do you have for us?
So I have an astronomy picture of the day from earlier this week, and there actually have been quite a few this week that were really cool, but this one gave me something to think about that I had never thought about before. It's called a full circle rainbow over Norway, and in the description, it talks about how a rainbow can actually be 360 degrees when viewed from the air. Perhaps Alan would know about that from his time flying. That had never occurred to me before. And this is a really beautiful picture of a rainbow in a full circle. That was just really cool and made me think about rainbows and rainbows in a way I hadn't ever done before.
It's actually two.
Yeah, I saw this picture. I like it.
So
it has
two.
to do with, the fact that there's a double one as well, but
Right.
it has something to do with raindrops. And then when it got to the point of saying it has a full diameter of 84 degrees, I just went, what? I couldn't make sense of that. And I just read on and tried not to worry about why it was 84
Actually,
degrees.
if you click on the author for the photo, you come across a photographer with some remarkable pictures.
Kathy, he was just Eucliding you.
Yeah. Okay, whatever.
Kathy, what do you have for us?
I picked one that's Frozen Soap Bubbles. It's a short Vimeo by Zaluska. I forgot to look up his first name, but you can go to his or her page, zaluskart.com. And this is just really cool. There's a nice little chime music playing in the background, but it's Frozen Soap Bubbles. Just amazing.
Frozen soap. Wow, that's
Very
beautiful.
cool.
Quite beautiful.
Yeah.
Nice.
Very cool.
Wow. It reminds me when I used to be on Vimeo. I had put some of my lectures there years ago, but I never went back. Rich, what do you have for us?
This is courtesy of McFadden. It's a New York Times article entitled "It's Turtles All the Way Down in the Fossil Record." Caught my attention because we've talked about turtles all the way down several times on TWiV. But this is a remarkable bit of serendipity because, believe it or not, there are scientific studies on the sort of structure and integrity of turtle shells that have to do with taking turtle shells and subjecting them to compression and finding out how and when they break and that kind of stuff. That gives you a number called the turtle compression index. And it turns out that there's a problem in paleontology where something that gets buried under sediment, it's very difficult to figure out when it was buried. You can't necessarily do it just from the depth of the sediment and something. And knowing when it was deposited gives you lots of clues about the history of that particular thing. So some clever individuals figured out that if you excavate turtle shells from the sediment, that the more apparently pressure that they've been subjected to, the more they're compressed. And so you can use the turtle compression index to date sedimentary deposits.
That's great.
Really?
Okay. And this
That's really good.
this article talks all about how to do that.
Nice.
Amazing.
Nice. All right, my pick, I was asked last week, Vincent, what are your top 10 favorite songs? And so I thought, well, this in the spirit of Dixon. I put together a list with the help of my daughter, and because she remembers what I used to like. And so I thought in the spirit of Dixon, I would share them. So you can make YouTube playlists, and all these songs are on YouTube. So here's mine. And the 10 songs, these are just not in any particular order. These are 10 songs I really like. I don't know if to my top 10, but they're 10 that I like today. Maybe tomorrow it would change. There's certainly other songs that I'd love to put in, but here they are. So first of all, it's Kind of Blue by Miles Davis, which I've always liked, and Dixon reminded me of it weeks ago when he picked this album. It's possibly one of the best jazz songs ever recorded. And the rest is really reflective of my youth as someone liking, rock and roll. So the next one is Linger by the Cranberries. Lovely vocals. Unfortunately, the lead singer died, but her voice is, she's Irish, and she has a lovely accent, great voice. And then one of my all-time favorites, Joni Mitchell, her song Free Man in Paris. It's a great song. Killer Queen by Queen. While My Guitar Gently Weeps with the Beatles, which, of course, has a guitar track by Eric Clapton, which is just amazing. Heart, Magic Man. We're talking with Dixon. They also did Barracuda, which is great, but I think Magic Man has got a really cool lilt to it. Then a band called Garbage, which the song I Think I'm Paranoid, which is just a wonderful rock and roll song. Arctic Monkeys. In my opinion, this band had one amazing album, and this is Do I Want to Know, which is from that album. This is a wonderful use of falsetto in this song. And then my favorite Lady Gaga piece, Poker Face. And then finally, the last one I actually discovered in the last couple of weeks, just poking around music on YouTube. This is by Carla Morrison, who's a Portuguese singer with just a lovely voice. The song is called Disfruto, and it's actually a remix of a piece she did earlier. If you go and look on her songs, you will find Disfruto, but it's much slower. This is a higher paced piece, a remix, and this is something very interesting that gets done a lot with music now. You will publish a song, and then someone else will remix it and redo it. They'll credit you, of course. And that really gives you a lot of variety in music. So those are my 10 that I really like right now. So enjoy
Vincent,
them.
I was working here yesterday on my annual report for the department, and I saw your pick and I put it up and I played through the whole list while I was working. It was really fun to hear.
I think they're all fun songs. Yeah.
Yeah. I'm going to have to listen again now with knowing your additional commentary.
Yeah.
So, yeah.
And it was fun putting together with my daughter. So we were sitting down, I think it was Christmas Eve. Yeah. I said, help me make this top 10. And she said, well, you like this and this and this and this. So I would put them in and I would take songs out. There are a lot of songs that went in and then came out because I had to limit it to 10. But I did want to have, at the minimum, Miles Davis up there. And, even though it's a recent addition, Carla Morrison, it's a fabulous song. It's got such atmosphere. And it just reminds me how much fun music is. I often don't listen to music. I don't know why, but I go through phases, and now I said, hey, I'm in the studio here. When I'm not recording or editing, I could play music. Why not? So I do. And
absolutely right.
So this is fun. But I can make another one too. So enjoy that. We have a listener pick from Charles, who wrote a letter. He's written many letters. He didn't have one today that we read, but he's written many letters over the years. Hello, Dixon. My ex-father-in-law was an excellent sax player. He would sometimes play with Vince DiMartino, the best trumpet player I have ever heard live. Vince was a local legend, both for playing and teaching. He may not make a top 3 or 4 pick, but it's well worth the time to seek out his music. Enjoy, and thanks for TWiV.
Yeah, so the pick that he gave to us is a really nice arrangement of MacArthur Park.
Oh, cool.
And so I listened to that too, with very good trumpet and sax.
Great.
That's it for TWiV 971, the last of 2022. You can find the show notes at microbe.tv/TWiV. You are welcome to send questions, comments, picks of the week to twiv@microbe.tv. And if you enjoy our work, you're also welcome to contribute financially to our projects here. You can go to microbe.tv/contribute. There are many ways that you can contribute, including mailing checks here at the incubator. I'm always excited during the day. I hear the mailbox bang. I put a mailbox on the door, a metal mailbox, so I can hear it bang, and I go and I find a few letters from all of you, and it's always fun to hear from you. So can mail. The address is at microbe.tv/contribute. Dixon Despommiers at Racaniello.org, thelivingriver.org. Thanks, Dixon, for another year.
You're welcome. It went by very fast, despite the trials and tribulations of all of us with our own individual problems. I just wanted to conclude my remarks this year by saying it's nice to see that we can all end on a high note.
Kathy Spindler's at the University of Michigan in Ann Arbor. Thanks for this year, Kathy.
Thanks. This is a lot of fun today, and it's been a lot of fun this year. I really appreciate everything you do to make it happen, Vincent. We couldn't do it without you.
I meant to say that too, of course.
Rich Conda is an emeritus professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you for the year, Rich.
Sure enough. Thank you. Always a good time, and Happy New Year, everybody.
Yes.
Brianne Barker is at Drew University, BioProf Barker on Twitter. Thank you for this year, Brianne.
Thank you for this year, Vincent and everybody else. I love having the opportunity to chat with you guys and learn and just have a great time. And I'm so excited to be a part of this.
Total.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology, the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. Have a happy new year, everybody. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next year. Another TWiV is viral.